So today we are going to talk about eukaryotic organelles. These are the small parts of a eukaryotic cell that are going to do some of the functions very much like the organs will in your body. They're going to allow a cell to be specialized and allow it to compartmentalize some of its actions so it is a more efficient cell and can do more things. When we talk about these organelles, remember these are all things that are going to be internal in the cell and are going to interact with each other in order to make the cell function. The first organelle that we'll talk about is the cytoplasm. The cytoplasm is the fluid that fills the cell. This is what gives it shape and holds all of the other organelles that are found. It's about the consistency of jello, so it's going to give some support to the cell as well as hold those organelles. Within that, you'll also see some protein fibers called the cytoskeleton. This gives it a little bit more support and also holds the organelles in place, keeps them where they need to be, or helps them move from place to place if they need to do so in order to facilitate the functioning of the cell. The cytoplasm is going to be the place where a lot of chemical reactions take place, and it's also going to be the site of some other processes that we'll talk about later on in the year. The cytoplasm is surrounded by the cell membrane, and that's the outer covering of the cell. The cell membrane is not typically talked about as an organelle, but just that outside covering of the cell. The next two organelles we will talk about, though, the cilia and the flagella, are extensions of the cell membrane, and they're going to be what provide movement for the cell or help materials move away from the cell membrane or away from the outside of the cell. They're made up of small proteins called microtubules, which just means that they're small tubes of protein. And then they're covered by the cell membrane or the plasma membrane to give them a covering. So it's not just a tube sticking out of the cell itself. The first one we'll talk about is the cilia. The cilia are very short projections. They're sometimes called finger-like projections from the cell. These finger-like projections are going to be used mostly for very small, precise movements or for moving particles around that cell or away from part of the cell membrane. You find cilia on cells that line, for example, the nasal passages. They're going to keep all of the cells lubricated by moving the mucus that's in your nasal passages, but not letting it get clogged up. So it's going to basically be what keeps the snot from running out of your nose all the time, but to keep all of the cells moist. You also find cilia in the digestive system to help move things through the digestive system, in addition to the muscle contractions of the digestive system itself. The other projection from the cell membrane are the flagella. The flagella are long whip-like extensions. They're almost like tails on cells. And they're going to be for these really big movements that move a cell from one place to another. Probably the thing you're most familiar with in terms of being a flagella is the tail on a sperm. If you've ever seen pictures of those, that tail on the sperm is the flagella, and it's going to help it move from place to place. Cells can have just one or two of these extensions, or they could have several up to many hundreds of the projections, depending on the type of cell and what it's going to do and what it needs to do. The next organelle that we're going to talk about are the ribosomes. The ribosomes are the organelles that are found in all cells, both prokaryotes and eukaryotes. So this is something common to both of them. And they're composed or made up of proteins and then a molecule called RNA. RNA is similar to DNA, and it's going to be a messaging type of molecule, and we'll talk about it more later on in the semester. The main purpose of having a ribosome is to make proteins. This is where your proteins are going to be made in the factories that put them together. So everything that gets made basically gets started here in the ribosome. These are found in all cells, like I said before, both prokaryotes and eukaryotes. They're found in plant, animal, viruses. Every type of cell has to have ribosomes. The next set of organelles that we're going to talk about make up something called the endomembrane system. The endomembrane system is a system of organelles that are all related to each other. They all have membranes, and they're actually all formed from pieces of the cell membrane that have pinched off or broken off and are used then to enclose areas within the cell. So because they're all made out of that cell membrane composite, and they all originate either at the cell membrane or at the nucleus, they're all considered to be related to each other. And in fact, we'll see later that some of the pieces of the endomembrane system can move from one organelle to another and just become part of that new organelle. So what's part of one organelle today may be part of another organelle and a different type of organelle later on. The endomembrane system includes the nucleus, something called the endoplasmic reticulum, the Golgi body, the lysosomes, and the vacuole. So all of these organelles, these 5 organelles, have the same outer composition or the same surrounding composition. What they do on the inside, depending on what they enclose, is going to be a little bit different. So what we're going to do is go through these organelles basically from the inside of the cell to the outside and talk about what they do and how they're related to each other. So the first organelle that we'll talk about is the nucleus. And what you see down here at the bottom is a picture of the nucleus with some details labeled for you. The nucleus has a double membrane, meaning it actually has 2 layers of covering. And through that 2 layers of covering, you do have pores or little holes. And those are there to allow things to move into and out of the nucleus. If you'll recall from your previous classes, the nucleus is kind of the control center for the cell. And so we've got to get information into the cell to find out what's going on in the cell, but then we've also got to get that reactive information out of the cell. And so you have to have these pores in order to do that. The nucleus is what contains the chromosomes, the DNA, and we'll talk about those more later on this semester, but they also contain an area called the nucleolus. It's this darker staining area, usually right in the middle of the nucleus. And this nucleolus is a place where you're going to find the synthesis of RNA and ribosomes. So we're going to make ribosomes here. This is the factory that basically makes the factory type of deal. And it's going to be important in terms of piecing together some of these really basic parts of the cell and making sure that we have the machinery in order for the cell to work. The next organelle moving out from the nucleus is the endoplasmic reticulum. The endoplasmic reticulum is responsible for synthesizing molecules and then moving them within the cell. So it's kind of like the roads in the cell. It's going to move material from one place to another. The endoplasmic reticulum is what we call continuous with the nucleus, meaning that it's actually connected to the nucleus, and the membrane of the nucleus actually just kind of outfolds and becomes the endoplasmic reticulum at a couple of points, and then it branches off from there. There are 2 types of endoplasmic reticulum, and those 2 types are dependent on what's associated with them and their function, and those 2 types are rough and smooth, and you'll often see endoplasmic reticulum abbreviated as ER. So when you see that capital ER, what we're talking about is the endoplasmic reticulum. So talking about these 2 types of endoplasmic reticulum and what they do, here's a picture of the endoplasmic reticulum, and you'll see here's the nucleus, and then it just becomes the endoplasmic reticulum. And you see the 2 different color variations here are to indicate the two different types, the rough and the smooth. They're not really colored differently, but it's easier for us to look at it that way. The rough endoplasmic reticulum, in this picture, it's shown closer to the nucleus. It may be further out. It just depends on the individual cell, has ribosomes attached to the surface. And because it has those ribosomes, those little pieces of protein attached to them, it looks like it's very rough, like it has nodules on it. And that's how the rough ER gets its name. And so it's going to be responsible for synthesizing and modifying and the shipping of proteins. Those ribosomes can attach to the rough ER, make those proteins, and then the proteins move within these passageways that make up the endoplasmic reticulum through to different parts of the cell. If the endoplasmic reticulum doesn't have any of the ribosomes on it, it's going to be called smooth ER. And ER is kind of interesting in that it can be rough for part of the time and then smooth for part of the time. It just depends on those ribosomes being attached to them. So the smooth ER is when we have no ribosomes attached to them. And when it doesn't have those ribosomes attached to it, the smooth ER is going to be responsible for synthesizing some molecules called lipids. Lipids are a class of molecules that are fats and oils and waxes. So basically, what we're doing is making fat with the smooth ER. And then also some of the carbohydrates that are going to be used. And carbohydrates are things like sugars and starches. These lipids and carbohydrates are going to be assembled in a particular way so that we can make more cell membranes. So the smooth ER is really where we're making the new outer covering for the cell as it grows, but also making some new organelle coverings with this. The other main function of the smooth ER, it's a very important function, is to break down toxins and poisons in the cell. So anything that could harm the cell, the smooth ER is going to produce the hormones that break it down or do the actual decomposition of those materials. So in cells that are going to be responsible for detoxifying your body or getting rid of things in your body, you're going to have a lot of smooth ER. For example, there's a lot of smooth ER in your liver where you're going to break down drugs and alcohol that are in your blood system, where you do some of that filtering. You've got this smooth ER to break them down. The next organelle you need to know about is the Golgi apparatus or the Golgi body, or sometimes called the Golgi complex. They're all the same thing. The important thing that unifies them as a single unit is this Golgi. The Golgi body was actually discovered by a man, an Italian named Golgi, and so his name is given to the organelle, but then you can call it the body, the complex, the whatever. But anytime you see Golgi, that's what we're talking about. And the Golgi body sits between the endoplasmic reticulum and the cell membrane. It's going to be kind of like the pathway as things move from the endoplasmic reticulum, then they'll move to the Golgi body and then out into the rest of the cell or out of the cell, depending on what's being moved. The main function of the Golgi body is to modify, package, and ship molecules. So what it's going to do is take the products of the endoplasmic reticulum, the proteins or the lipids or the carbohydrates that were made, and it's going to process them. It will repackage them. It might break them down or add them to some other molecules so that we get the right combination of things. And then it's going to ship those molecules either out of the cell or to other parts of the cell. One way to think about the Golgi body is to kind of think about it as like the pack and ship office or UPS or something like that. You take in your packages, they put them in a box, they get them all wrapped up, and then they send them somewhere. The Golgi body is doing the same type of thing, just in your cell. The next organelle that we'll talk about is actually something that pinches off of the Golgi. And when the Golgi has finished packaging all these products that it needs to do, it's going to form these little things called vesicles. They're right here. These vesicles then can move from place to place, and they get different names depending on what they contain. And so a vesicle containing one type of chemical is going to have a particular name or category, and a vesicle containing something else will belong to a different category. Um, The Golgi body can also synthesize parts of the cell wall around a plant cell. So we're still making all this stuff and moving it to different places so it can be assembled and used by the cell. One of the vesicles that breaks off of the Golgi body that you find in just about every cell is called a lysosome. And you'll see here down in the picture, here's the Golgi apparatus and then little parts pinching off of it, forming these lysosomes that are labeled in this cell. If you think about the word lysosome, it's kind of similar to Lysol. And that kind of gives us an idea of what we use it for, what cells use it for. Lysol is something we use to clean things with. It breaks down or disinfects things. The lysosome is actually going to contain digestive enzymes or things that are going to break down other molecules within the cell. And so you'll see that kind of cleaning up function with a lysosome. The enzymes that are found in the lysosome can be used to digest food within the cell. And so what they might merge with is a food vacuole. As you see in the diagram down here on the bottom, here's the food coming into the cell, being brought in and having a vesicle form around it. And then the lysosome merges with it and breaks down the food and becomes the digestive system. And then it can remove the wastes as well. So this lysosome can act as the digestive system for the cell. It's also going to digest or break down things as they wear out, other organelles as they wear out, things we don't need anymore because the cell has gone into a different part of its life, or something like that. So it can break down those as well. And then it can also aid in something called apoptosis. Apoptosis is programmed cell death. And as a cell ages, just like as any organism ages, it's going to start to break down a little bit more. And as it breaks down a little bit more, and these lysosomes are used more and more to break down those organelles that are wearing out, there comes a point in the cell's life cycle where the signal is given from the nucleus to go ahead and die, that we're going to break down this cell and use it for other parts. And so the lysosomes become the recyclers of cells as well. They're going to start to break down all of the cell parts, all of the organelles, and eventually break down the cytoplasm and the cell membrane so that it can be used as parts by other cells to rebuild themselves or to build new cells. So these lysosomes can be the recyclers for the cell. Another thing that can be formed from a vesicle that breaks off from the Golgi body is something called the vacuole. And the vacuole is simply a storage place. Vacuoles can be very, very small or they can be very, very large, and they're just going to hold things for the cell and keep them separated from the rest of the cell, kind of like a storage closet. Most commonly, they're going to store water or food, or they may store temporarily waste products before they're moved out of the cells. Vacuoles are found both in plants and animals, but their characteristics are a little bit different in terms of what they look like and where they're found in the cell. Plants, because they need to store a lot of water, have one really huge vacuole right in the center of the cell. And if you look at this diagram down at the bottom, you'll see here's the vacuole. And this is actually a picture of the whole cell, and it takes up most of the cell. That water vacuole is going to give structure to the cell in plants, and it's going to help the plant stay upright. When we talk about plants wilting, it's because their vacuoles are shrinking because they don't have as much water, and that outward pressure from the vacuole being there and pushing out on the cell wall allows the plant cells to kind of fold over on themselves, and so that's what you see with plants wilting. Um, so because plants have that one really huge vacuole, it's sometimes called the central vacuole. It can have smaller other vacuoles that store things, but it does have this one huge one for water. Animals, on the other hand, have a lot of smaller vacuoles. Um, sorry, have a lot of smaller vacuoles, and so they're not going to have that one big one. They're going to have little smaller size ones. Like these would be this about the size of vacuoles in animal cells. So these are all parts of the endomembrane system. Then we also have some organelles found in both plants and animals, well, and actually in all eukaryotic cells, that are more like bacteria than they are other organelles. And in fact, we think that probably at one point in time, these organelles might have been separate cells that were then just brought into a larger cell, and instead of digesting them, they used them to kind of help the cell work. The thing that kind of ties all these organelles together, other than being bacteria-like, is that these are the organelles responsible for energy transformations. They're going to do the conversion of food into energy, or from sunlight into sugars in plants. So they're all going to be related to this energy transformation. And then how they came to be in cells is what we call the endosymbiotic theory. Um, we think, looking at some of the fossils we have of cells and how cells probably evolved over time, that they might have been, these organelles might have been free living bacteria at one point in time, but then they were engulfed or brought into larger cells, but instead of digesting them, for some reason they weren't digested, and they actually helped the cell function because they were doing this energy transformation, this conversion, a lot better than the cell itself was. And so the cell, instead of digesting it, for some reason, was able to maintain those bacteria like organisms in its body. And then as the organism or the cell reproduced, those smaller cells got reproduced and passed along as well. And so we think that might be how they interact with each other. Some of the evidence we have for this is um, that these organelles, these energy transforming organelles, have their own set of DNA, and it's very different from the DNA that's found in other parts of the same cell. So the DNA found in these organelles controls just that organelle. These organelles divide on their own. They have their own independent ribosomes that function a little bit differently than the ribosomes in the rest of the cell. And so they're really doing their own thing. They're like mini cells within the cell itself. The one thing that makes them not those separate cells is that if you take them out of the larger cell, they won't live on their own for very long. And so there's been some type of relationship that's formed over time where these two types of um, cells and organelles depend on each other in order to maintain their life. So you have to have them together. They can't be separated anymore. Um, and then, like a normal cell, they do have multiple membranes. They have an outer membrane, and then they have smaller compartments that are surrounded by membranes, very much like having the organelle having organelles of its own. So once again, that's kind of that cell within a cell type of theory that we're talking about here. The two types of organelles that we'll talk about that fall into this category are the mitochondria and the chloroplasts. There are some other organelles that fall into this category, but those are ones you'll learn about either when you take Biology 2 or when you go to college and take some biology classes. These two are the ones we'll spend time talking about. So the first one is the mitochondrion. Mitochondrion is singular, mitochondria is plural, so you'll see it both ways. Make sure you know the difference. It is the same thing, just one's plural and one's singular. This is where cellular respiration takes place. And remember, we talked about respiration being more than just breathing. It's the actual use of oxygen or some kind of gas to break down molecules within the cell to release their energy. So that's what's going to take place here. So we're going to break down our fuel molecules to make energy, usually in the form of ATP. And these are found in all eukaryotic cells. Every one of them has them. Plants, animals, fungus, protists, they all have mitochondria. And it's very important that you realize that they all have them. A lot of times people think because of the difference between mitochondria and chloroplasts, only animal cells have mitochondria, but all cells actually have mitochondria. What you see in the picture here is actually a split picture. Part of it shows you a drawing of what a mitochondria looks like. That's over here on the left side. And then on the right side is actually an electron micrograph, or a picture that's been taken with an electron microscope that shows you what it actually looks like. Here's the outer membrane as it goes around. Here are some of the inner membranes and what would be analogous to the cytoplasm in the cell. And then over here you see the artist rendition of it, so you can kind of see why we think they look the way they do. But these are the powerhouses of the cell. You probably learned in middle school or elementary school that this is where the cell gets all of its energy from. And it's true. This is where we break down the food to get the energy so the cell can function. The next organelle we'll talk about is the chloroplast. It's the other energy transforming organelle. This one's found though only in plants because it's going to be the organelle that converts solar energy into chemical energy, where we're going to take sunlight and make it into sugars. So this is the site of photosynthesis, and this is a process we'll talk a lot more about later on this semester. And you do need to know that it's found in cells in plants and in some photosynthetic protists. But the thing you do need to remember is that not all plant cells have a chloroplast. If you think about this, it makes sense. Chloroplasts are the organelles that take sunlight and convert it into sugar. What parts of a plant don't get sunlight? Things like the roots, the internal cells, especially like in a tree, the cells that are under the bark, they don't get sunlight, so they're not going to have chloroplasts. Where you're going to find chloroplasts are in the leaves, on the outside edges of some of the other plant organs like the stems. Basically where you see green on a plant, that's where you're going to find the chloroplast. And like I said, it's going to be found just in those cells, not in every cell within a plant. So those are the organelles that are found in a eukaryotic cell. It's not a complete list, but it's a list of the most common ones and the ones you're most likely to see on the exams you take, whether it's an exam in this class or the end of course exam or a class later on in your career in biology. I hope you've enjoyed this, and that's the end of our lesson.